Okay, welcome back to another exciting uh, lecture learning. This one is going to be very different than before. We're going to work with a real biological data set, and I am not going to switch over to R, even though you're going to be required to do R for this lecture learning. That means I'm asking you to be a little bit more independent. As you've noticed throughout the course of the semester, I'm asking you to be more and more independent. And ultimately, something that's already happening, but what will happen with some students at least, is they will go off on their own path. The goal of the class is not to inculcate you to some kind of Ryan McEwen technique for R. It is to give you the tools and the empowerment to go your own way ultimately. And so this is part of the process. Also, I want to empower you by starting the class with a data set that's like, oh, LeBron James, but then increasingly delve into data sets that are more and more realistic, something that you might actually encounter if you're doing data analysis as a biologist. That's what you're going to see in this session. Okay. So in this lesson, we're going to work with a real biological data set from research. This data set has come from an experiment that I conducted in my research lab several years ago. The data set comes from an experiment in a stream where we were trying to show how different biofilms formed. Biofilms are the microbial community that assembles on substrate in a stream. How those biofilms accumulate in different kinds of stream habitats given different kinds of conditions. What we call a benthic habitat. Biofilms in a benthic habitat are microorganisms growing on the surface underwater. It looks like this. So specifically in this experiment, honeysuckle is a kind of a shrub. And we have streams where honeysuckle is growing over top of the stream. That's honeysuckle present. And streams where the honeysuckle is not over top of the stream, that will be honeysuckle absent. Here's an underwater picture of these two kinds of habitats. You can see the biofilm, and this is a really intense, this is like a kelp forest. Biofilms where the honeysuckle is absent, and this is in the same stream nearby where honeysuckle is present. Totally different kind of benthic environment. And actually the benthos, the microbial community in the stream, does a lot of the work in terms of nitrogen cycling and all the sort of the biology of the stream is very closely linked with those biofilms, including, by the way, wildlife, because you have small organisms that are use the biofilm as habitat, then bigger organisms eat those smaller organisms, and then bigger organisms eat that. Then you ultimately get to the macroinvertebrate community. The macroinvertebrate community then are like the insects. The insects then are fed on by fish, and then the fish are fed on by eagles. So this is like actually important to how the stream works from many different perspectives. So in this lesson, we're going to encounter what is a defining challenge of being a researcher in biology. What do we do when you have a small sample size? Remember that increasing sample size is a direct way to reduce noise in a data set. And remember also that a lot of biological data sets are noisy. There are many sources of variation in a biological experiment, including variation, or what we call error, from the experiment itself, like mistakes made by the experimenter, which is inaccurate measurements, or error that's inherent in the tools we use. If you use a ruler to measure something, the ruler is precise only down to the, whatever the smallest increment is on the ruler, for example. And how you apply that ruler might be different than the person, your neighbor. There's also variation, or what we sometimes call error, from the biological system. For example, organisms may have natural variation, such as genetic differences, that create variation in response to an experimental treatment. Environment may vary in some ways that create unaccounted for variation among the experimental units, something that you didn't measure. You weren't thinking about that. Ultimately, when we wish to calculate statistics to test our hypotheses, the variance becomes extremely important. There's a lot of variance. If there is a lot of variance in the system, then we are likely to fail to reject the null hypothesis, meaning we find no difference among the treatments in our experiment. There are a few basic ideas that underlie all statistics, which I have told you about already, but I'm going to give it to you yet again. We covered some of these earlier, but here we will review pertinent ideas to the upcoming analysis. Variance is key to statistics. And the numerator of our variance calculation here include the sum of squares, which I've described before. That effectively amounts to how much each value in the data set differs from the mean value in the data set. That's what this is here. Each value, and here's the mean. If you have very different values in your data set, you will have more variance. For example, the values in this little data set, A and B, to the right, have the same mean. The mean, the average is 10. But the individual values are much more variable in set B, in which each value, 0, 20, 0, 20, differs by 10 from the mean. The mean is 10. Each one of these differs by 10, either negative or positive. Where in A, each value differs only by 1 from 10. Okay, so subtract 10 from this, you get to 10. You add 1 to this, you get to 10. The numerator in the variance equation is specifically the sum of the variance as defined in this way. That is, bigger variance of each measurement from the mean is bigger variance in the calculation. More variance is more difficulty in discerning statistical difference. I know this is hard for you to follow. Also, this is just a really basic idea, but this thing right here ends up, if you go through and it's like, I'm going to open up the textbook and look. If you look at the basic math that underlies basically anytime you're calculating a p-value, this is showing up in there somewhere. So if you can kind of understand this, you can understand what's happening in data analysis, generally speaking. Sample size is also part of the variance equation. It is found in the denominator, which means mathematically, as you increase the sample size, the variance goes down. You want to have as large a sample size as you can. In biological experiments, sometimes the sample size is limited due to resources available or ethics or some other consideration. Many people in the class, if you're doing experiments right now, if you're like a graduate student, you're doing a chapter in your dissertation, you can't necessarily have 30 replicates in each of your treatments or something like that. There could be very many reasons why. It's going to mean we're going to have to kill hundreds of mice to do this experiment or we're going to have so many tanks are going to fill up the whole science center with fish tanks, or we don't want to have so many experiments because we don't want that much listeria running wild. There's very many reasons why, or there's not enough forests. So it's more common, and it's like the standard operating procedure if you're actually doing a hands-on experiment, to have a smaller sample size rather than larger. So this data set that we're going to analyze in the exercise, my laboratory had 5 different samples of stream biofilm in 5 different streams. We treated the samples as the experimental unit. Thus we have n equals 5 per treatment. Now we can argue whether this is pseudo-replicated or not. Let's have that conversation separately. For the purposes of what we're doing here, we've got 5 streams and n equals 5 in each treatment. We're going to calculate our variance and mean from those n equals 5 per treatment. We want to analyze the difference in biofilm using what's called ash-free dry mass, which we're not going to talk about anymore here. But just this is a measure of biomass, for example, how much biomass is this? And we're going to hypothesize that the reference site listed as ref will have more biofilm than other sites listed as mod and HVY, moderate and heavy. The reference sites, honeysuckle is absent, and HVY, honeysuckle, is present and very heavily invaded. It's not that important that you understand this design, but the biology of the system. But that's what's happening. So first, we're going to read in the data. and this is in the online repository and eCommons, which I've talked to you about before. You can literally go find this using this, and I'll paste the slides in as well, so you can click on this if you want. This should be a live link, or you can copy and paste it in, or you can just go on eCommons and find it and download. And here's the download data button right there. And if you right click on that, you can get the web address, and it's this. And so you can use biofilm equals read.csv and read it in this way. So pause the video and make it so. I'll literally paste this into the Isidore so you can just copy and paste it in. You don't have to type out all that. So go to Isidore, open the assignment. You already have Isidore open. So just go ahead and copy and paste this into your R Studio. And if you can't make that happen, I'm also going to do a write.csv and just paste the CSV file for you to upload. I don't want this to be the hang up in this assignment. Although it is fair to say that getting good at bringing data in is something you need to accomplish. You can't, by the end of this class, be like, well, I couldn't get the data to read in. That's just not a thing. It's no. You're going to have to figure out how to do it, okay? Otherwise, you're not empowered. It is not empowerment to be like, well, I couldn't get the data in. No. So do what it takes to practice so you make that happen. But in this particular case, since it's reading into a website, and I think there might be security issues or something in some of the, in some cases, I'll give you the CSV file. Okay, if you open up the data set, which you should always do, and take a look by just like typing, this is what we always do. Once the data are read in, or else if you just use that import, there's a tab at the top you can look at it. And I think you should always just type the name of the data set, highlight it, and run. And then look in the console so you can see the data set like this. If you do that and there's nothing here, you type it and you hit run and it says something, an error message, it says something like not found. Okay, stop. You can't run a data analysis on missing data. So you've got a problem. So you should always do that as just a standard operating procedure. You should see it in the console, not just in that tab. Okay, so if you open it up, you'll see that it's a large and complex data set. This is an experiment that was like conducted over the course of a year or more. And we can't analyze too complicated. But for the purposes of what we're doing in this exercise, we're just going to look at one date and one parameter. There's two parameters and many dates. So we're going to do a subset. And I've taught you subset already, but this is how we're going to do it. So this is our code. November 30 equals subset, comma, biofilm. That's the name of the data set that you've read in. Date equals quotes 11/30/2016 quotes close parentheses. Pause the video. Run the code. Okay, now we're going to work through this process, this ANOVA process, with the flow chart of best practices. Understand, as I've said before, if you read the literature, go online, whatever, you'll find many people taking many different pathways through this. And as I've already talked about in some detail, what I'm teaching you is an approach. I'm not saying it's the ultimate approach, the greatest. I'm saying it is an approach that is, I think, durable and literally publishable. It works. So this is a flow chart of best practices for the class. Now, we've talked about at great length. The first step in this is doves. So let's make a box plot. Here's the code I used. Pause the video and type the code. Got it? So you should see this. This is just a regular box plot. And here is a genome point plot. It's not a dot plot technically. I don't know what you call it. Point plot. I don't know. So here is the code. Pause the video. Type the code. I'm going to practice my drumming while you're doing that. Got it? Okay. So now it's time to assess distribution, outliers, variance, effects, sample size. If we overlay them this way, this is the code to overlay them. I think it's nice to have these separated. Here it is overlaid. Again, here's the code. Pause the video, type the code. Got it? All right. So now, distribution, outliers, variance, effects, sample size. So what's happening here? Well, distributions are pretty tight around the mean or median in all of these cases. I don't see, is this an outlier? Yeah, it looks like you've got a couple of outliers here, but they seem minor. Let's go back and look at this. Yeah, it doesn't look like, I'm not worried about those. Here's one and here's one. I'm not worried about those. They're pretty close. Variance, there's more variance in this data set than these, but it's higher as well. Just off the top of my head, I think we might have a problem with equal variance. Look how tight this one is. There's almost no variance. They're right on top of each other over here. In terms of effects, I went back to the whatever you want to call this dot or point graph or something. And we can see very clearly here, I just drew this line in PowerPoint, but you can clearly see that ref E and ref A look substantially higher than heavy, moderate. These are two moderate sites. Remember, there's 5 streams, heavy, moderate, ref. Reference has no honeysuckle. These all have honeysuckle. This has the heaviest invasion. Our hypothesis was the heaviest invasion would have the least, and the references would have the most. So this looks like what we were thinking. You go back to this graph. Again, from an effects perspective, these boxes look taller than all of these. The minimum point in the reference site is 0.02, which is higher than the highest point in any of these other sites. Very compelling from a statistical perspective. What about sample size? The last part of Doves. Okay, sample size. What's going on here? Here we've got n equals 3. This is barely enough. It's barely enough to calculate. You can see 1, 2, 3 clear here. 1, 2, 3. 3, 4, 5, 5, maybe 3. We have n equals 3 and 5. This is a small sample size. It is unequal. Right now, as a data analyst, from a Doves perspective, before we've done any tests, we haven't run any Shapiro tests and all that. We're going to do that. But right here and now, you should be thinking, we've got a problem. This doesn't look good. It's the scariest possible situation because you've got a hypothesis. It's a compelling hypothesis about the biology of the data. You've got a data set that seems to support the hypothesis. But before you run any tests, you're sitting here right now going, oh, this is, this is not going to work. Let's continue down the process. That's the end of Doves. Now, again, we're going to continue testing the data for assumptions. So now we're going to run the Shapiro test. This is the code for the Shapiro test. So we're going to go by November 30. That's the name of the data set, $. This was an open parentheses. AFDM, that's the ash-free dry mass, that's the column. November 30, $ code. Code is what we're doing it by, and then comma Shapiro dot test, close parentheses. So pause the video, run the code. Okay, got it? Here's what we've got. Okay, so again, this is just like from a snippets in. So this, don't pay too much attention to this. This is just trying to remind you that we're looking for normal distribution. That's what this does. It tests it. This is failing. This is failing. That is to say, this rejects the null hypothesis of a normal distribution. This one is negative 16. It's like 16 zeros. So it's way, way below 0.05. We have values indicating here that the distribution is not normal in multiple data sets. And this one down here is like really super negative, way below zero. But remember, the threshold value is 0.05. This is 0.000000016 zeros. So it's way less. Now in our process, I don't have creating histograms, but if this is for your thesis or something, you might do it. This is what the code would look like. I won't pause. You can pause the video to run the code. Again, it's not part of our formal process, but I would do this. This is ridiculous. Look at this. This is just an absolute. This looks bimodal. But again, when you're talking about an n equals sample size of n equals 3, how can you even have a distribution? You don't really. It's not even clear that you can even ask the question of the data set whether the distribution is here, because the sample sizes are too small. Look at this. What is this supposed to be? Ugh. So, all right, let's do the equal variance test. This is part of the process, so pause the video and run the code. It's bartlett.test, open parentheses, November 30, $AFDM, tilde, November 30, $code, close parentheses, run it. All right. Pause the video, run the test. Now you've got a p-value of 0.004695. This is suggesting that you do not have homogeneity of variance. Now, that's expected because the variance of this one is much greater than the variance in this one. And so, yeah, unequal variance. Oof, this is trouble. So we're going to do the test the assumptions graphically using this plot AOV command. By the way, there's another package I know some people have stumbled onto in the class that's a nicer way to do this. If you want to run it using that, go ahead. You don't have to do this. You can do it using the model test command. Again, if you don't know what I'm talking about, just ignore it. But some people do. But this is like the old school way or the base code way of running this. So it's par open parentheses MFROW equals C. That's going to concatenate 2. That's going to make up a stack of 2. So 4 graphs stacked. Then plot open parentheses AOV. That's ANOVA. Open parentheses November 30. That's the date. That's the data set. $AFDM. Tilde November 30. $code. Close. And run. Type the code. Run the code. I'll drum a little bit. I'll flip it over to the smaller one. This is what it looks like. So we're going to lay a pencil up here. It's a little swervy. By the way, this is another one we're not going to talk about, but just to say this should be flat. No. This is a different one, a leverage. This is looking really messy. We don't go into it, but this one's the only one I've really taught in the class, but it looks a little bit swervy there. So here's where we are. We've analyzed this step one with our Dove's assessment, Shapiro test, Bartlett test, AOV. The weight of the evidence from step one is that the data do not meet the ANOVA assumptions. That means we're going to come down this side of this flow chart. So we'll stop and say, again, this is a little bit advanced, but some practitioners would have you at this moment do some transformations. Okay, let's do log transformation or square root transformation. If you don't know what I'm talking about, just ignore this. But there is a school of thought that what you do is then transform the data to try to force it to meet the ANOVA assumptions, so you can come down this path. I think that's perfectly good practice. A lot of people do that. I'm not teaching it in this class because it's got its own problems. Like, if you're going to start running through a whole series of transformations to try to force your data to meet the normal assumption, that seems a little sketchy in its own way. Like the pathway I'm teaching here is heading down a non-parametric. So you just say, I'm just going to analyze the data without the assumptions. That has costs, pluses and minuses, versus transforming the data to try to force it to meet this. That also has pluses and minuses. And I think the transformation approach is too complicated for the class. So this is a way that many people approach their data. It's just like, okay, it doesn't meet the assumption, I'm going to use a non-parametric test. Kruskal dot test. So we're heading down. This is for the purposes of our class. We've done step one. Now we're going to do step two. The weight of evidence is that the data do not meet the ANOVA assumptions. We saw that in the Shapiro test. We saw it in the Bartlett test. We saw it in the strip chart and box plot. We're heading down this path. The next step is Kruskal dot test data measures data group. So it looks like this. The non-parametric ANOVA. Non-parametric means we're not worried about whether it meets the assumptions. So type the code, run the code. Okay, Kruskal rank sums test, p-value 0.002929. Hey, we're saying that the data do not meet the assumptions, so we're running the Kruskal rank sum test, and we got a statistically significant result. This is rejecting the null hypothesis of no difference. This result of the test matches what seems to be the case on this here graph. You just look at it and go, these are higher than those. This looks like what we should find. So from a science perspective, we're okay. The Kruskal test brought us forward. Now, the next thing we have to do is to do a pairwise comparison, because this is testing the overall model effect. And here we have this other break between whether it's significant or not. It is significant. It's less than or equal to 0.05. So we're heading down this to the pairwise comparison part of the flow chart. So this is the end. And for there, we're going to use this pairwise.wilcoxon.test. This is, again, a non-parametric test. You're saying here, I understand that this is not meeting, that the data set does not meet the standards for running a normal ANOVA. It does not meet the ANOVA assumptions. So we're going to do this pairwise test. You have to adjust the p-value. There are many different ways, and I'll in some code. In the code I'll show you that if you want to like check out the different p adjustments. This is a good one though. This Benjamin or Benjami or whatever, I don't know how to say that, and Hochberg BH adjustment. It's going to show you the differences, the pairwise differences. So we can do this one to the next one to the next one to the next one. So here's the code. Type the code. Pause the video. Here's what we've got when you do that. First of all, a heap of warning messages. It's giving you the results here in this little matrix, which is comparing heavy B to moderate C, heavy B to moderate E, heavy B. So it's going like this. Then, or whatever ones are not, yeah. So it's going to do in the pairwise process we've talked about before, and it's presenting it in this little matrix. However, R is also saying, basically like, hey man, hey y'all, this is messed up. Your data set. That's how I interpret this. It's like, oh, this is not. This does not look good. Which is what you would expect. I just showed you before. You've got n equals 3 and n equals 5. It's like a weird, bad situation. Right. What are you going to do now? Okay, so here's like, let's just stop and think about it for a second. So in the field, you have good evidence of an effect, but the analysis procedure looks like a mess, and none of the treatments meet the 0.05 threshold. So you're really messed up here. You've got a significant overall effect, that's what this is, but literally no individual differences. That's a really weird place to be. I should stop and just say something. Let me go back to the graph here. Sorry, I didn't say this before. You may be asking yourself, and this would be late. Should have asked already. I should have brought it up already. What the hell's happened? What kind of experiment are you running that has an n equals 3 and n equals 5 over here? What, didn't you just say that you have n equals 5? Here's the problem. When we put these experimental units out, which were actually bags filled with marbles. So we put like little bags in the stream. It's like a mesh bag. In the bag were marbles. The marbles were the substrate upon which the microbes are colonized. And then you sonicate the marbles to get the biofilm off and then do the analysis. We also had a molecular analysis to like actually ask what microbes are present. So we put 5 bags in every stream. And then you go pick them up at intervals because they literally sit in the stream for a certain period of time so the microbes can colonize. So why would you have 5 in one stream and 3 in the next? This is stupid. The answer is we put 5 in every stream, but the damn raccoons came and tore them apart. Possibly in some of the streams, specifically this one, Ref E, was also a place where children sometimes showed up. There was like a swing set not far from there. And so kids, we think, came and were like, oh, this is fun. And they took the bags from my experiment and threw them at each other and threw them like into the trees. we found bags, our experimental units like in trees and stuff and torn apart. So yeah. Raccoons maybe are children. There was mammalian disturbances. And let me just also pause to say, I told you this was over the course of a year. November 30, 2016 was the best one. That was one where some of the dates we went out the stream, like April or something, there were none here. They're gone, all gone. All the marbles were thrown like on top of the picnic shelter and stuff. They were just ripped apart or gone. We never found them again. So you don't know what's happening. Nonetheless, when you're trying to do the analysis now, the experimental units, the bags are gone. Why am I telling you this? Is this a tangent? No, this is not a tangent. Please stop assuming that you know what a tangent is. You don't. If you think this is a tangent. This is actually the core, right down the middle. Because for you to be empowered, you need to hear this and understand that this can happen to you. Your experiment can be ruined in this way. For me, I'm a professor. If this experiment gets ruined for me, if you're a master's student and your experiment gets ruined in this way, you've got a big question mark now as to whether you just spent 3 years for nothing. This is not a tangent. This is talking about the reality of trying to do analysis of biological data. And what is the risk when you have n equals 5 experimental units and you've got a situation where you might lose some of those experimental units. This could be mice in a tank or fish. If you've got, this could be whatever experiment you're running. And so we have to live with this now because it's too late. The kids have thrown the marbles at each other or threw it with the car truck by. They're gone. Now back to the results at hand. So in the field, if you stand there and look at the stream with this underwater camera, you've got really good evidence that when honeysuckle is removed, you've got a totally different microbial community in the stream. It's a very cool scientific result. Not only that, you've got evidence from the statistics, at least for that one date, that there is an overall effect, but now we can't tell them apart. So what can you do? If you were stuck so what I did as a professor, because I just left it, and the data set's unpublished, it's just posted there, and it never will be published. It's just a research pathway that I didn't go down. It's lost. If you had to do it, and it was your master's thesis, and you're sitting here like, dog, am I gonna have wasted these years? And you feel like you absolutely have to do something with the data set. There's no off ramp. Here's what I would do. You're the researcher, so you can define what you consider significant. In biology, we use P equals 0.05 as a critical value, as a standard operating procedure. You could say, hey, because of the issues we've had, I'm relaxing that to P equals 0.1 would be okay. That's like a 10% chance. And if you do that, then now you've got something here. You go, okay, so go through these values, 0.69, 0.69, 0.69, 0.071. These are just outside the critical value of 0.05. So if you do that, you could map on here A, because heavy B, so you go heavy B, moderate C, the same. Different. And then C, actually ref E, is different also from ref A. See that? These two are different from each other, and ref A is also separated from everything. So this is what it would look like, the letters. You have to understand that at this point, you're more or less SOL by design. That's what this is. Ryan McEwen was SOL by design. However, if you have to make something out of it, this is what you can do. So we have an unsatisfying statistical analysis. If this is your experiment, you would be frustrated. It seems like you have a strong experimental result. It looks like the ref A and ref E are higher than the others, but the statistics indicate that things are very unclear. These are my data, and yes, I am frustrated. I think this would be a really cool paper that would get cited by a lot of people, and also tells us something about the biology of the stream that's really important. But SOL by design. I had a small sample size. I thought reasonable replication would be n equals 5, but there were problems in the field that caused me to lose experimental units. We think raccoons came, or little kids, and they, had equivalent of a snowball fight with my experimental units across the stream, or whatever. Nonetheless, we got there, gone, or shattered, marbles everywhere. So we had very low sample size, in some cases none. And the data set I showed you November 30th, it was an equals 3 on one site. So yes, SOL by design. This is the reality of working with biological experiments, and it is not uncommon for something like this to happen to you. We have a couple of options. Maybe you could do a permutation test or transformations. I mentioned that before. It's questionable though. You've got the small sample size. You can't change it. It is what it is. If I wanted to pursue this scientific idea more, I need n equals 10 probably in the streams, and I might need to think about is there some other way to protect the experimental units? Do I need to put a little fence or something up, or what's the way I can protect these? So and probably as a researcher, since I'd never done an experiment like this, it probably would have been smart to do a pilot study. Just go out there the year before I ran the experiment, and just put some bags out to see what happened. That's a smart thing to do. Pilot studies are very smart. If I put n equals 10 out there and I lost half, I would still be okay. Like if raccoons take away half the sample size and I'm left with n equals 5 in each treatment, okay. But I can't, now it's too late. And I'm not doing the experiment again. But yeah. If you have to write up the results as they are, and it's your thesis at stake, what you can do is something like what I'm about to do. It's not a final figure, but you can make some figure like this, make it look nice. And you could write something like this. Our experiment suggests that Lonicera macchiae, that's honeysuckle, Lonicera macchiae invasion intensity influences stream biofilm biomass. This is honeysuckle intensity, and this is biomass. Figure 1, refer to this figure. Results of the non-parametric Kruskal test suggest a significant overall effect of invasion intensity on biofilm ash-free dry mass. AFDM P equals 0.003. This is the overall treatment effect. Remember, go back up. That's this. Overall treatment effect. Results of the non-parametric Kruskal test suggest a significant overall effect of invasion intensity on biofilm ash-free dry mass, AFDM, semicolon, P equals 0.003. Pairwise comparisons via the Wilcoxon procedure, however, yielded marginally significant results, suggesting that biofilm AFDM within ref A, the ref A site, was higher than all other treatments. This is the highest. And ref E was higher than the moderate or heavy site. That's what C is here. But lower than ref A. All results P equals 0.007. So I rounded up and rounded down. I just put P equals 7. As described in the method section, the loss of experimental units due to wildlife activity, I should say mammalian activity, because it could have been kids, but maybe wildlife is probably good. The loss of experimental units due to wildlife activity potentially influenced our capacity to statistically discern differences among treatments. This is a very good sentence. A reader of this sentence understands what's happening. As described in the method section, the loss of experimental units due to wildlife activity potentially influence our capacity to statistically discern differences among the treatments. Now, especially not this, if you put the plot with the points on there, instead of this is just a box plot. If you did the the graph we did before that has the points as well, so they can see the sample size, and you write it up this way, your hands are clean. There's nothing fake about this. You've owned it. You've owned your data analysis. The reviewer of your paper can look at this and go, okay, I believe it or I don't. And you can move forward. And the fact that you use P equals 0.01 as a critical value, yeah, all right, fine. Let the reader decide. So that ends the session on small sample size ANOVA. I hope you learned something here. That's the purpose of this. And yeah, we'll see you in the coding club